When you think of a mushroom, you probably picture that familiar cap and stem, maybe sitting quietly on the forest floor. But what if I told you that iconic structure is just the visible tip of a vast, hidden biological iceberg?
It really is. It's a truly profound concept once you start thinking about the actual scale of the organism beneath what we normally see.
Yeah. So today we're taking a deep dive into one of the most fascinating and, well, diverse groups of fungi, the Basidiomycota, specifically the order Agaricales, what we commonly call mushrooms.
Right, the guild fungi and their relatives.
Exactly. And our mission here is to unpack the dense science of these organisms. We're talking structure, reproduction, their surprising roles out there in the environment.
And even their direct impact on us, human health, culture, all of that.
We've got this fantastic chapter from an Introductory Mycology textbook. It's packed with rich details, deep insights, really good stuff, and we want to give you a shortcut to understanding these essential life forms.
Connecting the complex biology to why it actually matters,
Yeah,
Maybe arm you with some mind-bending facts along the way.
definitely. So let's get into this hidden world. We mentioned that mushroom, the cap and stem, the basidiocarp, you called it. That's not the whole organism, is it?
Not even close, really. It's just the reproductive part, the fruiting body, as we often say. The actual fungus, the main body, is usually hidden from view.
Okay.
It's this extensive network of really delicate thread-like structures. We call them hyphae. And altogether, they form this massive web called the mycelium.
Mycelium, right.
Yeah, and this mycelium network is often underground or maybe weaving through decaying wood. It's doing most of the organism's work absorbing nutrients, growing.
Wow. And it's funny, the word mycology itself, the study of fungi.
Oh yeah, the etymology is great. It comes straight from the ancient Greek myx, which literally meant mushroom.
Yeah.
So the visible part gave the whole field its name.
That's brilliant. So the mushroom we see is like the apple on a huge unseen tree. Okay, let's talk anatomy then, the parts we can see. You got the cap
The Peleus, yeah.
and the stalk.
The stipe.
Right. But under the cap, that's where it gets really key for identification, isn't it?
Absolutely critical, because that's where the spores are made. You'll usually find either these thin radiating plates,
Yeah.
we call them gills, or sometimes lamellae.
Like little curtains hanging down.
Exactly. Or in some groups, like the boletes, you won't see gills. Instead, it's like a layer of deep or shallow tubes all packed together, and you can see the openings, the pores.
Ah, okay. Like a sponge layer underneath.
Kind of, yeah. And those surfaces, the gills or the tubes, that's where the spores are produced and eventually released.
And here's something that always amazes me.
Mm.
How mushrooms seem to pop up overnight. One day there's nothing, the next day, bam, a whole bunch of them.
It looks like incredibly fast growth, doesn't it? But it's not actually rapid cell division, which is what you might think.
Oh, so what's happening?
It's actually the astonishingly swift enlargement of cells that were already there inside a tiny little preformed structure, like a little knot, called a primordium or button.
So it's like inflating a balloon really quickly.
Precisely. It's driven by something called turgor pressure. Basically, water pressure inside the cells pushes them to expand rapidly, and this force is incredible.
How incredible.
Incredible enough that some mushrooms can literally push their way up through hard-packed soil or even asphalt.
Through asphalt. No way.
Yes, it really makes you appreciate the power packed into what seems like a delicate structure.
That's amazing. Okay, I've also heard about veils on mushrooms. What are those about? They sound kind of mysterious.
They are important clues. Many young mushrooms have protective coverings. There can be an inner veil, which is this thin membrane stretching from the edge of the cap down to the stalk.
Protecting the gills when they're young.
Exactly. And as the cap expands, this inner veil tears. What's left behind is often a ring of tissue on the stalk called the annulus.
Ah, like a little skirt.
Yeah, or sometimes it's much more delicate, almost like a cobweb. Then we
Right.
call it a cortina.
Okay, annulus or cortina. Got it. Is that the only veil?
Some mushrooms also have, or only have, what's called a universal veil. This is a layer of tissue that completely encloses the entire young mushroom, like it's inside an egg.
Like an egg. Wow.
Yeah. And as the mushroom grows and bursts out, this universal veil ruptures. It often leaves a cup-like structure at the very base of the stalk called the vulva.
The vulva. Okay.
And you might also see remnants of it left as scales or patches scattered on the surface of the cap.
But those little flaky bits on some mushroom caps or that cup at the bottom, they're actually leftovers from this veil.
That's exactly right. And these structures, the annulus, the cortina, the volva, the patches on the cap, even though they can be fragile and disappear, they are incredibly important taxonomic characters. They give mycologists vital clues for identification.
So don't ignore the little scraps of tissue.
Definitely not. And if we dive even deeper, looking, say, with a microscope, there's a whole microarchitecture inside.
Go on.
Well, that spore-bearing layer on the gills or tubes, that's called the hymenium. It's packed with specialized cells, primarily the basidia, which are the actual cells that produce the spores.
Basidium.
Yep.
Okay, makes sense for Basidia mycota.
Right. And the tissue within the gills themselves, the stuff holding it all together, the hymenophoral trauma, its structure is also key. Is it made of similar hyphae, or does it have these distinct spherical cells called spherocysts mixed in that help separate major groups, like the Russelaceae family?
Wow, okay, so the details just keep getting finer and finer. Let's switch gears slightly. We know how they're built, but how do they actually reproduce? Tell us about the fungal cycle of life.
Okay, so the cycle typically starts with a basidiospore. Think of it like a seed, but it only has one type of nucleus, what we call homokaryotic.
Okay, one nucleus type.
This spore lands somewhere suitable, germinates, and grows into a primary mycelium, that network of hyphae again. But this primary stage often doesn't last long or do much on its own.
So what needs to happen?
Two compatible primary mycelia need to find each other and fuse together. This fusion of their cytoplasm and hyphae is called somatogamy.
Somatogamy. Body fusion, kind of.
Pretty much. And the result is the dikaryotic secondary mycelium. Dikaryotics meaning two nuclei. Each cell in this secondary mycelium now contains two distinct compatible nuclei, one from each parent. And this is usually the main, long-lived vegetative body of the fungus.
So most of that hidden network, the mycelium, is actually dikaryotic, with two different nuclei coexisting in every cell. That's wild.
It is. They live together, divide together, but they don't fuse. Not yet.
So when do they fuse? The actual marriage of the nuclei?
That happens much later, and only within those specialized cells in the hymenium, the basidia. Inside the basidium, finally, the two nuclei fuse together. That's karyogamy, nuclear fusion.
Karyogamy, okay.
And immediately after that fusion, the now diploid nucleus undergoes meiosis, the special type of cell division that reduces the chromosome number back and produces typically 4 new basidiospores, each ready to start the cycle again.
Wow. That's quite a journey from spore to spore. A complex dance.
It really is. And while that sexual cycle is dominant, I should mention some can also reproduce asexually, maybe through simple fragments called oidia, or tougher, thick-walled resting cells called chlamydospores. But sexual reproduction is the main event for diversity.
Okay. Now, for someone trying to identify mushrooms they find, you mentioned spores, there's a practical technique, right? The spore print.
Oh, absolutely vital. A spore print is basically just collecting the spores that the mushroom releases.
How do you actually do one? I've tried it. Sometimes it works. Sometimes not so much.
Yeah, it needs a little care. You basically cut the stipe off right near the cap. Then you place the cap, gill side or pore side down, on a piece of paper. Using both black and white paper side by side is best, so you can see light spores on the black and dark spores on the white.
Good tip. Then what?
Then you cover the cap with something like a bowl or a glass to keep the humidity up and prevent drafts. Leave it for a few hours or maybe overnight. When you lift the cap carefully,
You get a pattern of spores.
exactly, you get a pattern showing the arrangement of the gills or pores, and crucially, you see the color of the spores in mass.
And that color is really important.
Hugely important. Spore color is a major identification key. They can be white, cream, pink, yellow, ochre, various shades of brown, purple brown, black, even olive green.
A whole rainbow, almost.
Pretty much. And beyond the color, under a microscope, the spore size, shape, and especially the surface texture ornamentation, we call it, and there are like 12 different categories for that, are all taxonomically critical.
Wow. Any other spore tricks?
Mycologists also use chemical tests. Meltzer's reagent is a common one. It's an iodine-based stain. Depending on the species, spores might turn bluish-black, which we call amyloid, or yellowish to reddish-brown called dextrinoid, or show no reaction, another layer of clues.
Amazing how much information is packed into those tiny spores. Okay, let's broaden out again. Structure, reproduction. Now, what about their ecological roles? They're not just sitting there, right? They're doing important jobs.
Oh, absolutely fundamental jobs. Many agarics are saprobes. That means they are decomposers. They break down dead organic material, fallen leaves, logs, dead trees, even animal dung.
The great recyclers.
Exactly. Without fungi doing this, nutrients would stay locked up in dead stuff and ecosystems would literally grind to a halt. They're completely essential for nutrient cycling.
And sometimes this decomposing activity, this hidden mycelium, leads to things we can see, like those fairy rings. My grandmother used to tell me stories about those.
Aha, yeah, they definitely capture the imagination. Seeing mushrooms pop up in a circle in a lawn or field, it looks magical.
But there's science behind it.
Oh, yes. It's actually the result of the mycelium of the fungus growing outwards from a central starting point, like ripples in a pond, but underground. The mushroom fruiting bodies then form at the outer, active edge of this expanding circle where nutrients are probably most available.
And I've heard the grass inside the ring can look different.
That's often true. Especially in older rings, the grass inside can be greener. That's thought to be because as the older mycelium in the center dies and decomposes, it releases nitrogen, fertilizing the grass.
Huh. So the ring leaves a trace even without the mushrooms.
Yeah, and some of these mycelial rings can be incredibly large and astonishingly old. The mycelium of Marasmius orades, a common fairy ring mushroom, has been estimated to live for up to 400 years in some cases.
400 years. That's an underground empire right there.
It truly is.
Okay, so decomposers, ring formers,
Yeah.
but they also form partnerships, right? These mycorrhizae things.
Yes, mycorrhizae. This is absolutely crucial. It means fungus root, and it's a symbiotic association between fungal hyphae and plant roots.
Symbiotic, meaning both benefit.
Typically, yes. It used to be described almost as a reciprocal parasitism, but we now understand it's usually mutually beneficial and incredibly widespread. It's the rule in nature, not the exception. Many scientists believe most plants couldn't survive in the wild without their mycorrhizal partners. Some even argue that the colonization of land by plants way back when was only possible because of these fungal partnerships.
Wow. So what does the fungus do for the plant?
A lot. The fungal network acts like a massive extension of the plant's root system, vastly increasing the surface area for absorbing water and nutrients, especially things like phosphorus and nitrogen, which can be hard for plants to get on their own.
So better food and water uptake. Anything else?
Yep. They can also help plants tolerate environmental stresses, drought, extreme temperatures, heavy metals in the soil, and they can even protect the plant roots from attack by pathogenic fungi or bacteria.
That's a huge package of benefits. What does the fungus get in return?
It gets sugars, carbohydrates, and maybe some vitamins that the plant produces through photosynthesis. Things the fungus can't make itself. So it's a fair trade, usually.
And you mentioned there are different types of mycorrhizae.
Yes, two main types we should know. First, there are endomycorrhizae. Endo meaning inside. Here, the fungal hyphae actually penetrate into the cells of the plant root.
Inside the cells. Wow.
Yeah, they form these characteristic structures inside the root cells, balloon-like things called vesicles, and these incredibly branched tree-like structures called arbuscules. That's where the nutrient exchange happens. This type, also called VA mycorrhizae, is super common, found in most crops, garden plants, grasses.
Okay, so endo means inside the cells. What's the other type?
The other major type is ectomycorrhizae. Ecto meaning outside. In this case, the fungal hyphae form a dense sheath, like a thick sock, around the outside of the root tip. We call this the mantle.
So it doesn't go inside the cells.
It sends hyphae between the root cells, forming a network called the Hardig net, but it doesn't penetrate the cells themselves. This type is really common with trees, pines, oaks, birches, beeches, eucalyptus. Many familiar forest mushrooms are the fruiting bodies of ectomycorrhizal fungi.
And this has practical uses too, right? Like in forestry.
Absolutely. Ectomycorrhizal fungi are hugely important in reforestation. Seedlings grown in nurseries are often inoculated with specific fungi before being planted out, especially in difficult sites. It dramatically increases their chances of survival and growth. It's applied mycology in action.
That's incredible. Nature's partnerships, harnessed by us. Are there other, maybe more unusual fungal relationships out there?
Oh, definitely. Fungi get into some really specialized gigs. For instance, think about certain termites in Africa and Southeast Asia. They literally farm fungi.
Termites farming fungi.
They cultivate the mycelium of fungi from the genus Pyrenomycetes inside their nests on special combs made from chewed wood and termite droppings. The termites eat the mycelium, and the fungi get a protected, stable environment. And the mushrooms that pop up near the termite mounds, often prized edibles for humans too.
Wow, termite farmers. Anything else?
There are also the Atine ants, the leafcutter ants in the Americas. They have a similarly ancient and sophisticated relationship, cultivating specific fungi in their underground gardens on chewed-up leaves. They completely depend on their fungal crop.
Ants farming, too. Amazing. Any other surprises?
How about predatory fungi? The common oyster mushroom, Pleurotus ostritus.
I eat those. They're predators.
In a way, yes. Besides decaying wood, its hyphae can produce these tiny, sticky droplets that trap and paralyze microscopic worms called nematodes in the wood. The hyphae then penetrate the nematode, kill it, and digest it.
No way. Oyster mushrooms eat worms.
They do. They can also apparently parasitize and consume bacterial colonies. It's a reminder that fungi have diverse nutritional strategies.
Mind blown. Oyster mushrooms are metal. Okay, one more weird thing. Some mushrooms glow in the dark. Foxfire.
Yes, bioluminescence. It's true. Certain species, like the jack-o'-lantern mushroom, Ascoloculares, or sometimes just the mycelium of fungi like the honey mushroom, Armillaria amelia, can emit a faint, eerie glow. You might see it in rotting wood at night.
Why do they do that? Is it to attract insects or something?
That's the mystery. The textbook basically says, of what use this would be to the fungus is not known. Just adds to their mystique, doesn't it?
It really does. Okay, let's pivot to where fungi most directly intersect with us. Human impact. Food, medicine, but also peril.
Yeah, this is critical. There's definitely a growing interest in foraging for wild mushrooms. It can be rewarding, but
There's a huge but.
a massive one. Let's be absolutely clear. There is no simple test to tell if a wild mushroom is edible or poisonous. None. Forget the myths about silver coins tarnishing or peeling caps or insects eating them. They are all dangerously wrong.
So what is the safe way?
The only safe way is to learn to identify individual species with 100% certainty. And if you are a beginner, you must have your finds checked by a recognized expert before even thinking about eating them. The stakes are just too high.
Learn your species, get an expert check. Got it. But cultivation is much safer.
Oh, absolutely. Globally, about 14 species are cultivated commercially, maybe 6 on a really large industrial scale.
Like the ones we see in the supermarket.
Exactly. Things like Enochitok, Flammulina velutibase, often grown in jars or bottles. Shiitake, Lentinula edodes, traditionally grown on logs. Oyster mushrooms, Pleurotus ostritus, grown on straw or other agricultural waste. And, of course, the king of cultivated mushrooms.
The white button mushroom.
That's the one. Agaricus brunescens also includes portobello and cremini, which are just different strains or stages of maturity. It dominates the global market.
How do they grow so many of those?
It's a pretty sophisticated industrial process, actually. It starts with producing high-quality spawn that's the mycelium grown on sterilized grain. This spawn is then mixed into carefully prepared compost, usually based on horse manure and straw.
Okay.
The mycelium grows all through this compost that's the spawn run. Then they add a top layer of soil or peat mix called the casing layer. This change actually triggers the mycelium to start forming mushrooms.
Ah, the casing induces fruiting.
Right. And then growers meticulously control temperature, humidity, and carbon dioxide levels to manage when the mushrooms appear, usually in waves called breaks or flushes. It's very controlled agriculture.
Wow. Much more complex than I realized. Are they actually good for you, nutritionally?
Yeah, they stack up pretty well. They have a high nutritional index, apparently better than most veggies except maybe spinach and soybeans. Good source of minerals like potassium, phosphorus, and various B vitamins. Some vitamin D, too, especially if they've been exposed to UV light.
And what about medicinal uses? I hear a lot about that, especially from Asia.
That's true. In some cultures, particularly China and Japan, certain mushrooms like shiitake, reishi, maitake, they're highly valued for supposed medicinal or tonic properties. Claims range from boosting the immune system, having anti-tumor or antiviral effects, lowering cholesterol, promoting longevity, even acting as aphrodisiacs.
That sounds incredible, but
But, and this is a really important but, as the textbook cautions, not all claims relative to the medical properties and other merits of mushrooms are supported by scientific data. So a healthy dose of skepticism and looking for rigorous scientific evidence is definitely warranted.
right. Approach with caution. Now, the dark side. Mushroom poisoning. This is serious stuff.
Extremely serious, and one genus stands out for causing the vast majority of fatal poisonings, especially in Europe. Amanita.
Amanita, I know that name.
Yeah. Species like the pure white destroying angel, Amanita virosa, and the sort of olive green cap, death cap, Amanita phelloides. Beautiful, but absolutely deadly. They account for something like 90–95% of mushroom fatality.
What makes them so lethal? What's the toxin?
The primary culprits are compounds called amatoxins. These are incredibly potent toxins. What they do is inhibit a crucial enzyme in our cells, RNA polymerase II. This enzyme is essential for making messenger RNA, which means cells can't produce vital proteins.
So it shuts down protein production.
Essentially, yes. And this leads to cell death, particularly affecting organs with high rates of cell turnover, like the lining of the intestine and, critically, the liver.
And the poisoning has distinct phases, right? Which makes it even more insidious.
Exactly. It's terrifyingly deceptive. Phase 1 is the latency period. This can last 6 to 12 hours, maybe even longer after eating the mushroom. During this time, the person feels perfectly fine. The mushroom might even have tasted good.
So you don't even know you've been poisoned yet.
Right. Then comes phase 2, the gastrointestinal phase. This hits suddenly with severe, violent vomiting, diarrhea, and abdominal cramps. Dehydration becomes a major life-threatening risk at this stage.
Okay, awful, but at least you know something's wrong.
Yes, but then comes the cruelest part. Phase 3, the false recovery. After maybe 24 hours of gastrointestinal misery, the symptoms can subside. The person might feel better, think they're over the worst of it,
Oh no.
but they're not. The amatoxins absorbed earlier are now silently, relentlessly destroying their liver cells.
And that leads to the final phase.
Phase 4. Liver failure progresses, often accompanied by kidney failure. Jaundice sets in, confusion, coma, and death typically follows 6 to 8 days after eating the mushrooms, sometimes longer. It's a slow, agonizing process. And crucially, because of that latency period, by the time severe symptoms show, the toxins are already absorbed and treatment becomes incredibly difficult. There's no single, universally effective antidote.
That is truly chilling. And again, reinforces never guess with wild mushrooms.
Never. And those folk tests? Useless. Even the Meixner test, sometimes mentioned for amatoxins using newspaper and acid. It's not reliable for edibility testing by amateurs. Don't rely on it.
Okay. Are there other common poisonous mushrooms people should be aware of?
Definitely. One common one causing lots of gastrointestinal upset, though usually not fatal, is Chlorophyllum molybdites. It's often called the green-spored lepiota and pops up in lawns and grassy areas. Looks a bit like some edible species, but check the spore print. It's greenish.
Green spore is bad news in this case.
Generally, yes for that one. Another interesting one is Coprinus atramentarius, the common inky cap. It's actually edible unless you consume alcohol anywhere near the time you eat it, even a day or two before or after.
What happens then?
It contains a compound called coprin, which blocks the metabolism of alcohol at an intermediate stage, causing really unpleasant symptoms, flushing, nausea, palpitations, similar to the drug antabuse, which is used to deter alcoholics.
Wow, nature's own antabuse. Okay, shifting from deadly to different. Hallucinogenic fungi. These have a long history too, don't they?
They certainly do. The most well-known are probably species in the genus Psilocybe. They often have this characteristic bluing reaction if you bruise or handle them.
Ah, yes, the magic mushrooms.
Right. They contain the psychoactive compounds, psilocybin and psilocin. There's strong evidence of their use going back centuries, particularly in Mexico, where they were considered sacred or divine mushrooms used in religious ceremonies.
What kind of effects do they cause?
It varies, but typically distortions of perception, touch, vision. Colors might seem more intense, objects might seem to breathe or move. Mood can shift dramatically, euphoria, anxiety, deep introspection, sometimes fear or panic, a profound alteration of consciousness.
And what about the really iconic one, the red one with white spots?
Ah, Amanita muscaria, the fly agaric, instantly recognizable. Famous in folklore illustration.
No, but actually used to kill flies.
Apparently so, traditionally broken up in milk to attract and stupefy flies, but its main fame comes from its use in shamanic rituals in various cultures, particularly in Siberia. There's also that controversial theory by R. Gordon Wasson that Amanita muscaria was the original soma, the divine plant mentioned in ancient Hindu texts.
That's fascinating. What are its active compounds? Is it the same as psilocybe?
No, completely different chemistry. The main psychoactive compounds here are ibotenic acid and its breakdown product, muscimol. They affect different neurotransmitter systems in the brain. It also contains muscarine, which causes physical effects like sweating, salivation, blurred vision, but usually not in high enough amounts in A. muscaria to be life-threatening on its own, unlike in some other small poisonous mushrooms where muscarine is the main toxin.
So, with both psilocybe and Amanita muscaria, given their history, should people experiment with them?
The textbook strongly warns against it, and for good reason. Using these as recreational drugs is really risky. Why? Because the effects are highly unpredictable. They depend massively on the dose, which can vary even within mushrooms from the same batch, but also on the individual's personality, their mood going into it, and the setting where they take it, the set and setting. A bad trip can be terrifying and psychologically damaging. It's not something to be trifled with.
Good warning. Okay, last section. Let's take a quick tour through some key fungal families within the Agaricales just to get a feel for the diversity. Taxonomy is always changing, right?
Oh, constantly. Especially now with molecular DNA techniques. We used to classify fungi based purely on what they look like: gills, pores, veils, spore color. Now, DNA analysis is revealing some really unexpected evolutionary relationships,
Like what?
like finding out that some gilled mushrooms are actually more closely related to puffballs or stinkhorns or even underground false truffles than they are to other gilled mushrooms. It's forcing a major rethink of the fungal family tree.
Wow. Okay, so bearing that in mind, let's look at a few big families. Boletaceae, the boletes.
Right. These are usually easy to spot because, as we said, they have tubes with pores underneath the cap instead of gills. Many are mycorrhizal with trees. A key feature for ID within boletes is often whether the flesh changes color when you cut or bruise it, sometimes dramatically blue or red or black. That can be a clue, sometimes indicating a poisonous one. And as mentioned, DNA shows this family is broader than we thought, now including some gilled fungi and other forms too.
Okay, how about Russelaceae?
Ah, these are interesting. They include Russula and Lecterius. A defining feature is their texture. The flesh is often distinctively brittle, snapping, kind of like chalk, due to those spherical cells, the spherocysts, in their tissue.
Brittle, and Lecterius does something weird.
Yes. Lecterius species are often called milk caps because when you cut or break the gills or flesh, they bleed a liquid called latex. It might be white or orange or yellow, even blue or purple, depending on the species. The color and how it changes is a super important ID feature.
Bleeding mushrooms.
Oh.
Cool. What about the huge family Tricholometacea?
Yeah, traditionally this was a massive dumping ground family, and it's being broken up a lot by molecular studies. But it contains many familiar names. We mentioned the cultivated shiitake and inokitic, also the fairy ring mushroom, Marasmius, the bioluminescent jack-o'-lantern, Omphalodus, and the honey mushroom, Armillaria, which is a major forest pathogen causing root rot known for its tough, black, shoestring-like strands called rhizomorphs that spread underground. And don't forget the predatory oyster mushroom, Pleurotus.
Quite the rogues gallery in there. Then Amanitaceae.
Back to Amanita. Contains some prized edibles, like Caesar's mushroom, but also, as we stressed, the deadliest mushrooms known, like the deathcap and destroying angel. Defined by those veils, often leaving a vulva and an annulus and typically white spores, such a potent reminder of the duality in nature.
And Agaricaceae, where the button mushroom lives.
Exactly. Home to the genus Agaricus button mushrooms, portobellos, and many wild relatives, often with pinkish then brown spores and an annulus, but also includes that green-spored lawn troublemaker, Chlorophyllum molybdites, shows how even within a family you get edibles and poisonols.
Copernaceae, the inky caps.
Yep, the genus Coprinus and its relatives. Their defining feature is that amazing self-digestion process called deliquescence. The gills and cap literally liquefy into a black, spore-laden ink as they mature. Very distinctive.
Truly unique. Strophariaceae.
This family is notable because it contains many of the hallucinogenic psilocybe species, often characterized by that bluing reaction and typically having dark purple-brown spores.
And lastly, Cortinariaceae. You mentioned this was huge.
Enormous. The main genus, Cortinarius, is the largest genus of mushrooms in North America. They get their name from that delicate, cobweb-like partial veil, the cortina, that covers the gills when young. Many are mycorrhizal, often brightly colored, but identification is really difficult. And unfortunately, many are poisonous, some dangerously so, like Cortinarius oranus, which contains a slow-acting kidney toxin. Another group requiring extreme caution.
Wow, what a journey. So wrapping this all up, what's the big takeaway? We've gone from hidden networks to fruiting bodies, reproduction, decomposition, symbiosis, food, poison, medicine, ritual.
I think the takeaway is just the sheer complexity, importance, and often hidden nature of this kingdom. They're fundamental to how ecosystems work, from breaking down the dead to enabling plants to grow. They interact with us in profound ways, both beneficial and dangerous.
Yeah. So next time you see a mushroom, maybe poking through pine needles or even pushing up sidewalk crack, remember it's just the visible sign of this ancient, vast, and frankly, quite mysterious network operating all around us.
Absolutely, and makes you wonder, how much more is going on down there that we don't know about? What other secrets are these silent organisms holding?
A great thought to end on. Thank you so much for sharing all this incredible insight today.
My pleasure. It's a fascinating world to explore.
And thank you all for joining us on this deep dive into the world of Agaricales. Keep learning, stay curious, and we'll catch you on the next deep dive.